finish up, in theory, Chapter 4, though I'm probably going to try and drag my feet a little bit, because Chapter 5, first off, we're just going to kind of skip over. I've got one slide on Chapter 5, and we're just going to briefly talk about it, and that's it. Chapter 5 is largely lab techniques, which none of you will ever have anything to deal with, so it doesn't really make sense to spend a lot of time talking about it. Chapter 6, if you thought any of these chapters were a beast so far, Chapter 6 and 7 are a really rude awakening. Okay? So I would highly recommend that you read those chapters very, very thoroughly prior to coming into class. I personally will have to read them again very, very thoroughly to make sure we're super clear on that. What we're looking at within 6 and 7 is that oh so fun section of kinetics, okay, and complex kinetics. Those of you that went through 152, it's nothing in comparison to the kinetics that we end up dealing with. Okay. The interpretations that come out of that, we will greatly simplify. Okay. So we're going to boil that down to pretty much being able to look at a graph or a curve and say, okay, that means it's this, that means it's this, okay, without having to deal with the mathematics, which is unfortunate because you got to deal with a little bit more memorization, but that's kind of what biochem is. We ended with our secondary plus structure last week. So we're looking at individual motifs. So you hear this word motifs pop up every so often. Right? All it's referencing is that there are some particular structural patterns that get repeated in a bunch of different proteins. The primary, try to avoid saying primary in this context, the largest motifs that you could find for secondary structure are your alpha helix and your beta sheets. So those are the big ones that you're responsible for. You'll see these other ones pop up every so often. We've got on the extreme left a beta barrel, and actually kind of in the middle too. Okay. Because you're looking at beta sheets, or a beta sheet that is folded up on itself like a barrel. Okay. And the beta barrel we can kind of see being a secondary motif because it's still a beta sheet, but it's kind of folded on itself, so it's a little weird. The last one in that case, which I don't even remember what to call it, over here is still classified technically as a secondary structure, even though we really very clearly have different motifs bound in the same thing. Okay, we have the alpha helices that are coordinated on the outside of a beta barrel. Okay, that's why you run into this kind of gray area terminology, and they call it secondary plus, because it's not quite secondary, but it's also not quite tertiary. Because our tertiary is the official step up, where we now have all of our secondary pieces, those bonding motifs, are now interacting with each other. And that's where you run into an issue. If you have a beta barrel and alpha helices, you think that's tertiary. Okay? Tertiary as a class is more everything, the entire protein coming to its full structure. So we can kind of dodge our alpha helix beta sheet issue by saying there's a lot else left with that protein. Because there's a lot else that's not described there, it's not officially the tertiary structure, it's officially the secondary structure. Okay, so it's a weird gray area. So secondary is parts of your structure folding. Tertiary is the entire protein, okay, that entire big piece. Okay, so that's going to include the secondary motifs that you see and how they interact with each other. That's going to include the rest of the protein as well. Okay, stuff that didn't fold nicely into neat little patterns. These get separated into two main categories. Okay, so now we're looking at the larger protein. You get these fibrous proteins and you get globular proteins. Okay. Most of the proteins that we tend to think about or work with tend to be the globular proteins and less so the fibrous proteins. Any ideas why?
ATP?
No, ATP synthase is just an example. So we've got a couple examples underneath these. So an example for a globular protein, ATP synthase is globular. The fibrous proteins, collagen, fibroin, keratin.
The globular proteins are what is actively interacting in your body.
No, not quite. It's not a bad idea, and it kind of has to do with location. It really has to do with this last thing on there.
Water soluble?
Water soluble versus water insoluble. Pretty much everything we're working with is trying to extract things out of a water environment. It's pretty easy to pull our proteins out into a water environment and then try and study them. It's not so easy to pull water insoluble proteins out, because if we try and pull them out with, say, an organic solvent, well, then we destroy their tertiary structure and they don't fold appropriately, and we're not really studying the appropriate protein. So the fibrous proteins can be a little bit more challenging to work with because of that. So a lot of that has to do with our solubility. So in general, your globular proteins are water soluble. Let's just go ahead and call that H2O soluble, and in the other ones, they are water insoluble. And those are just kind of your general trends, kind of your true-false situation between those two states. So how do we go through and end up with these two interactions? So what we're concerned about is how does the secondary motifs come together to form your tertiary structure? So what different interactions come into that? What becomes important? What doesn't become important? Okay. So your book has a fairly nice figure that shows a nice little ribbon representing our protein, and then how that ribbon can potentially interact with each other. So in that first section, what do we have? Right here, what do we have? We've got an alpha helix, followed by a beta sheet. It's a little bit weird because they're showing all of these lines. What are those lines supposed to represent? Hydrogen bonding. Why hydrogen bonding? So an interaction is true as well. We can be more specific with hydrogen bonding. Why hydrogen bonding?
Because hydrogen bonding that hold the backbone.
Kind of, sort of. Hydrogen bonding is what holds together beta sheets. Okay, it's the hydrogen bonding within the backbone. So those lines are supposed to represent hydrogen bonding. Okay? We could get really nitpicky on this just because I happen to vaguely remember this. Remember there were two types of beta sheets? And the hydrogen bonds either form nice regular ladder-like forms, or you had those kind of slightly angled ones. Do you guys remember which one was which? Well, I knew anti-parallel and parallel too, but I don't remember which one's which as far as the stacking.
Anti-parallel is, yeah, and the
Oh,
anti-parallel.
well, this
is
would
where
have
it
to
is
be
the
anti-parallel.
crisscross, because they don't line up.
Yeah.
Yeah,
They don't want that?
yeah, they don't line up. They're like opposite of
They're
each
like
other.
off. So we could try and look at the hydrogen bonding in that case to decide. I would argue that we shouldn't be looking at the hydrogen bonding. What kind of beta sheet do we have here? Parallel or anti-parallel? How do you know it's anti-parallel?
Yes, the direction.
Protein has to fold back. For it to be parallel, it would have had to loop back around and come up the same direction. Okay, so we've got an anti-parallel beta sheet, regardless of how those, whoops, that's not supposed to be alpha. Anti-parallel. And yeah, that's the exact figure I was thinking of, Teresa. So we've got those bonding motifs, and now how those interact with the rest of the protein chain is ultimately where we get into our tertiary structure. So what they're showing by this figure is all these individual pieces that could come into it. So the next thing that comes along, I know this might be difficult to see, but what we've got in this little corner is a metal. And in particular, it's a metal with a +2 charge. Okay? So when we move up into tertiary structure, something else that can contribute to that binding is now metal ions coming in and coordinating. Okay? Well, what would the metal ions coordinate to? What charge is the metal ion? Positive. Okay. Our backbone is involved in our secondary structure, so it can't be secondary structure, or it can't be our backbone that's binding to the metal. So if it's not the backbone, it's more than likely the...
Chain.
What do you mean by
Side
chain?
groups. The other side groups.
Okay, we're looking at our R groups,
Why can't?
or our
mine be
side...
the backbone?
Our backbone is primarily involved in the secondary structure. So when we move up to looking at our metals, we're going to be looking at anything, and we'll talk about this in just a second, something that can coordinate to the metal. We're going to list off what things are most likely to coordinate to the metal here in just a second. Okay, what would we expect a characteristic of whatever that was to coordinate to the metal? What charge was the metal? Positive. So what charge should the thing coordinating to the metal be? Negative. While there are a source of electrons in the backbone, is there a better source of electrons found in the R groups?
Yeah.
Good answer. Like what?
Like the acids.
Like all of our polar acidic side groups. Because all the polar acidic side groups at pH 7 release their hydrogen and they become negatively charged. Okay? So what are our polar acidic side groups?
Cool.
So that's looking at the group, but what's the name of your...
Glutamic acid.
You've got aspartate and you've got glutamate. So a few people have asked about this. So let's go ahead and address this now. Glutamate versus glutamic acid. Okay, or for that matter, aspartate versus aspartic acid. What's going on with those? All you're referring to is the charge of that R group. In the aspartate and glutamate, you're looking at the negatively charged carboxylate salt. And the glutamic acid or aspartic acid, you're looking at the hydrogen still being on the structure. So you will see glutamate and glutamic acid used interchangeably as far as nomenclature goes in reference to amino acids. Okay, so be aware of that. All that is looking at a different protonation state. Should they be calling it something new and different? No. But it's probably a holdover from real nomenclature systems from organic chemistry. Okay. So our negatively charged amino acids are going to be the most likely to coordinate with metals. Okay. So if we looked at our metal site, the little dotted lines are probably an interaction coming from aspartate or glutamate because those ones are negatively charged. Are there other sources of electrons that are not negatively charged? Yeah, okay, there's a couple other sources. You could look at the backbone. Okay, the backbone tends to be buried inside the structure, which makes it difficult to get access to. But if we were looking at a bend in the protein, that is possible that the backbone could do some other interactions. Okay, what were you going to suggest?
No, I was talking about the basic group.
Got to be careful. Our basic side groups are going to be a little bit trickier to work with. If we change the pH appropriately, okay, we can make those basic side groups have lone pairs that can potentially interact. But I'd argue before we even get into the basic side groups, we have another option. We have our polar neutral side groups. Okay, your serines, okay, your cysteines. Those have electron pairs that could potentially donate out to a metal and help kind of coordinate it. Okay? So what we're looking at is what things can interact to generate those. Next interaction on that list. I have to erase some of this stuff. What's that next interaction look like? I know those of you sitting up front have a easier time of this.
Hydrophobic.
Okay, it's being labeled as hydrophobic. Well, what's happening in a hydrophobic interaction? Okay. Not water soluble. So what amino acids might we find in this not water soluble situation? All of our non-polar R groups. Okay, so examples you could pull right off the slide. Is there a BE? What the heck? Oh, isoleucine. So you got leucine, valine, isoleucine, glycine, alanine. all of those you would typically find in kind of this hydrophobic pocket. Okay? So what's happening in that situation is, remember, we're looking at something that is water soluble, or not necessarily water soluble, but in an environment that has a lot of water. So what are those R groups going to do? They're going to get as far away from the water as possible, okay? Which means they tend to fold into these pockets where we get these hydrophobic interactions. So those would be all our non-polar R groups. Another interaction that we can pull out of this would be disulfide bonds. Those of you with curly hair or have ever had a perm, guess what's disulfide bonds? Or guess where disulfide bonds are typically used? It's very common in curly hair. Okay? Those actually form an actual covalent bond between R groups that then forces a particular kink in the overall structure. Hence, curly hair works really well for that. Okay? So how can we form that disulfide bond? Well, we need sulfur groups. Do we have an amino acid that has sulfurs? Yeah, what was that? You've got cysteine. So, did I spell that right? I think I spelled that right. You've got two cysteines that have actually reacted with each other and formed that covalent bond, forcing a kink in the structure, okay, or forcing a kink in that protein. After that, we start to drift off into some more interactions. Some of those may be more familiar. Okay. What we're listing there is side chain hydrogen bonding. And it doesn't have to be relegated to just side chain hydrogen bonding. It can be solvent side chain hydrogen bonding. Okay, we've got a lot of polar groups. Those polar groups are going to orient themselves to kind of minimize that charge on them through hydrogen bonding interactions. Okay, so we got hydrogen bonding. I guess I can underline that. And then the last possible interaction that we could pick up would be what they're labeling as our electrostatic, but we've also referred to as an ionic interaction. Anything where we have charged species interacting with each other that is not a metal interaction, we can classify as electrostatic. Okay? Why not the metal interaction? Because we've subcategorized that as a special situation, okay, as metal coordination, as opposed to just electrostatic interactions. So we've got all of these things potentially contributing to our tertiary structure. Okay, so it's kind of neat that all of these things can work together. What makes this even more fascinating is that our proteins will do this all on their own to fold to their natural state. Okay? So all of these kind of interactions sum together to get us to our native state that's super reactive and does what it needs to do. And if we change any one of those interactions, or potentially two or three of those interactions, we nullify that protein's ability to fold into this tertiary native state, and it can no longer react and do what it needs to do. Okay. There's a little bit of debate behind this, or this next kind of question, but which of these interactions is the most important to protein function?
Hydrophobic?
What was that?
Hydrophobic?
Why hydrophobic?
That was one of the answers on the. I guessed on that, but I don't know. Because it's
Okay,
the
this is
one that's repelling.
where the debate kind of comes from. All of your hydrophilic interactions can interact with water. So all of those we would expect to be located where on the protein? On the outside, because that's typically where our water is. Where's all the hydrophobic stuff? On the inside. Well, if we ignore the inside for the moment, and just look at all those hydrophilics, those could extend outwards and fill and interact with water all the way around. What is preventing them from extending it outwards? It's the inside hydrophobic amino acids that are pretty much folding up and repelling away from water, causing the structure to ball up. Okay? So there's kind of a balance or a dynamic between these, either your hydrophobic or your hydrophilic interactions contributing to it. And it's not strictly as simple as just saying, oh, it's just this or it's just that. Okay? But the hydrophobic tends to be the one given the most weight to your tertiary structure and your overall bonding. All right. The next thing I kind of want to address with this is you might see why Chapter 3 would become important, and when I talked about what you needed to memorize as far as amino acids. I said you should probably get one from each group. Okay? This is your primary reason why you need one per each group. Because I could go through and say, given a hydrophobic pocket, give me an amino acid that would fit in that pocket. Okay? Given metal coordination, what amino acid would you likely find coordinating to that metal? Okay? So having an understanding of what amino acids are available and where they exist within your tertiary interactions can be pretty important, and it's a very easy test question that kind of gets at all of the things that we've been talking about. Okay? So be aware of that as a possible question popping up. So we got all those fun interactions listed out again. Questions? Man, that was 30 minutes on one slide.
So, I mean, do you only have the hydrophobic interaction in between, I mean, inside the protein? What about the other bonds, like the sulfide bonds and the metal bond? Are they found outside or inside?
So they can be found pretty much anywhere, okay, depending on what's actually happening within the structure. And depending on where the protein is located. So that's another thing that I kind of disagree with our statement that we made earlier on just globular proteins being water soluble and the fibrous ones being water insoluble. Because if we go back and look at the structure for the ATP synthase, you had a top part that was outside the membrane. Absolutely water soluble. But there was an inside part. Where was that inside part found? Inside the membrane. Well, what's inside the membrane? Hydrophobic stuff. So one of the things that made trying to solve that structure so difficult was that half of the molecule was water soluble and half of it was very insoluble. But the overall thing is still referred to as globular. Okay. So depending on where your protein is located within the cell, be it in the cytosol or be it just hanging on to something on your membrane surface or being embedded in the membrane, you're going to get varying interactions and varying qualities that come out of it as well. Other questions?
So those electrostatic attractions aren't just, they're just any amino acids basically? It's just the amine and then the carboxylic acid of those?
Careful. Can any amino acid give me the amine shown or the acid shown?
Probably not.
So we're looking here, and we're looking here. Can any amino acid give me those two interactions? No. If I give you this amino acid, I'm being lazy with the abbreviation there. This amino acid would not give me the interactions found there. Why not? Let's get into the idea. What does this green thing represent? A big fancy. The backbone. So if we look at the end of this backbone, what do we see right there? A C for, no, not quite carbon. The carboxylate end. And if we look at the other end, what do we have at the other end? You have the amine end. If we go back and look at that electrostatic interaction, that is an NH3 plus. Where is that NH3 plus found? Yeah, it's found in the side chain. That NH3 plus is part of the side chain, not part of the backbone. Okay. So it's not any amino acid, because if we take a look at the amino acid that I provided here, that carboxyl and that amine get bound into the backbone of the protein, which means they disappear from my interactions. I no longer have a C or an O minus and an NH plus. Okay. What I have is that R group, that CH3. So what amino acids can contribute that NH3 plus think you're right. Lysine. I think it's supposed to be capital. Is it arginine? Histidine. I thought there were. Is it just 3? Okay. What amino acids could contribute that COO minus? All of our acids. So we've got our aspartate, GLU, right? GLN is the glutamine, I believe, right? Okay. So those electrostatic interactions, while theoretically could be your amine end and your carboxyl end of your protein, are quite likely not those two. It's very likely looking at the R groups within your amino acids. Okay. Other questions? Okay, so just like you did in organic chemistry, we now have to kind of predict or see this tertiary structure. So there's a variety of ways that we could go through and do this. You can do 2D NMR. So if you guys remember HNMR from organic chemistry, if we look really carefully along these axes here, you'll notice that we're listing a chemical shift in parts per million. We're looking at the range roughly between 7 and 10. Why do we not go lower than 7? It's not pH. There's no hydrogens down there in that chemical environment. All of our hydrogens are shifted up into the chemical shift range between 7 and 10 for this case. So we don't care lower and we don't care higher. You notice if we take a look at the other axis, we can now start to see how those interact with these. So we're looking at a 2-dimensional NMR, or you can even shift it up to a 3-dimensional NMR. And what you analyzed in OChem was a 1-dimensional NMR. Okay, so in this case, what we're now saying is where we see these dark points, let's pick one, say here, is that the hydrogen found at 10 ppms is interacting with a hydrogen found at roughly, what is that, 6.5 ppms. Well, for them to interact, they have to be close to each other. So what does that mean? Well, I now know that a hydrogen that shows up at 10 needs to be interacting with a hydrogen that shows up at 6.5. I can now start to pick some information about where my protein is all folded together. Okay, when you looked at HNMR in organic chemistry, probably the largest compound that you saw was maybe a molecular weight of 200 grams per mole, somewhere in that range. That's probably on the big end. When we look at our proteins, remember one of our relatively small proteins had 396 amino acids in it. The molecular weight is significantly higher than 200 grams per mole. Okay? So it becomes a lot more of a challenging process to go through and process these things and put it together and then be able to interpret how it comes together. Okay? There's lots of active research in this, and we aren't going to spend any time talking about trying to interpret those because it is way too difficult. Okay? The other thing that we could go through and do is look at X-ray crystallography. So if everything works perfectly, what you can potentially do is take your protein and crystallize it. Right? Just like you did in recrystallization experiments in organic chem. You get nice, neat, beautiful crystals. Well, that works for small organic compounds with relatively uniform intermolecular forces. The protein is not small or neat or uniform. So it usually takes a while to actually crystallize a protein. But once you've got a perfect crystal, you can take it in and fire X-rays at it and you can generate an X-ray crystallograph. Based off of the dot patterns that come out of the X-ray crystallograph, you can then interpret that into structure. And you can determine the actual structure, 3-dimensional structure of your protein. Kind of cool. Okay? Also, not the easiest thing to discuss. Okay? Which means that what we're really getting to is, this is neat techniques. If you moved into a biochemistry field, not even a class, into a biochemistry field, you would probably look at these aspects. Okay? I had a friend that got his PhD, and his PhD was crystallizing a single protein and coming up with a structure. That's what his PhD was. Okay? I've got another friend that was working on spider silk, and she did NMR on spider silk to determine its 3-dimensional structure. Okay? So things that we would think are relatively straightforward, or at least seemingly simple, just what is the structure, turn into big PhD projects. Okay, so it's kind of a neat process. I'm hoping that this is going to link out to a neat video. Yeah, it is. Neat. So what this is looking at is probably somebody else's PhD project.
So are the groupings, sorry, back up.
No, it's okay.
The previous slide. Are the groupings where it starts to fold? Where you have
What?
your line? No, on that one.
These groupings?
Yeah, correct.
I actually forget. I believe that's a primary section on where you're getting a lot. No, that's duplication of it. So you see where it overlaps? They're overlapping at the exact same points. That's the self-replication, not self-replication, but overlap of the data. So if we take the two-dimensional model on that x-axis, we look at the two-dimensional model on our y-axis, 10-10, okay, it's the exact same amino acid. So we would expect to see a nice sharp line running through the middle of the data where it's exactly matched, okay, because it's self-referencing it's effectively. We're saying on one thing, there is a peak here for this amino acid. If we now look at the other axis, there's a peak there for that exact same amino acid. So it's going to double that intensity. We'll see that line. What we're looking at is off that axis, how do we then develop what things are close to it? So we could then say, okay, 10, I don't really see any interactions with anything else. Those are probably found on the outside of the protein, potentially. I'm actually kind of just guessing here. Those sections don't have a lot of interactions or secondary interactions with other amino acids, which is why there's nothing out there. If we move further in, you'll notice that at 8, we see a lot of different peaks. That's because whatever shows up at 8 is interacting with a lot of other amino acids. So we're seeing a lot of those kind of secondary signals pop up. I do feel a bit bad about this because I haven't had time to go through and find some better references on myoglobin and hemoglobin. It is a massively important topic for any of you moving into further health fields. So you should be reviewing this section very, very carefully. The homework has a lot of questions on myoglobin and hemoglobin for that very reason. And they're all spun in slightly different formats. A lot of graphical interpretation. Okay? So spend some time to try and interpret those graphs and figure out exactly what's happening in those situations. Because what they're trying to do is take the information discussed in the textbook and kind of textual matters and turn it into something more interpretive of what you can get out of it. Okay? So our classic tertiary example is myoglobin. So myoglobin, we got a bunch of random facts for it. It's one of our compounds that can hold oxygen. Okay, so it's a good protein for oxygen, not transport, grabbing, oxygen storage. Thank you. So what we've got is the overall protein structure in green, and then in the middle you see this little red section. That little red section is a heme group. Okay, and the heme is really what's responsible for holding on to the oxygen. Okay, the protein is what holds the heme and then facilitates the ability for that oxygen to either be bound tighter or looser, because if the protein comes in and adjusts where the oxygen exists, it can kick out the oxygen. It may not be super obvious to see at first, but if we go through and look at the structure for the heme group, what we're looking at is a porphyrin, or a pseudoporphyrin, with an iron atom in the middle. So this is why iron is super important for oxygen transportation. So that iron sits in the middle and can coordinate to the oxygen. This is going to be awesome to attempt to draw this in 3 dimensions. So let's say we've got our oxygen molecule kind of bound in here. Well, if that oxygen is then coming out of the plane at us, okay, is held up here. Remember, the heme is now holding on to our oxygen, and then the protein is then holding on to the heme. Well, if the protein comes by and has a giant piece of something, okay, say an aspartate ion, okay, aspartate because it's got that O minus, and that now sits in front of the iron. Can the oxygen molecule get in? Well, no, the aspartame's blocking it. So what that protein can go through and do is adjust what amino acid sits in front of the iron to either facilitate oxygen binding or prevent oxygen from binding. So one of the things that we have to remember is that our proteins are dynamic. They are constantly moving. It's not just frozen in place. So with that constant motion, we can either facilitate oxygen binding or not facilitate it by blocking the binding site. Okay. So with myoglobin, oh man, I just went through and answered that question. So why do we have the protein at all if the heme works? Because hemes do bind oxygen relatively well. Why use the protein at all? Okay. Heme is perfect at binding oxygen. What do you mean inhibit? We don't want oxygen just stuck to the heme. We want oxygen being released in our cells so that it can be consumed and used in our reactions. So we do want oxygen to be able to bind because we need to get the oxygen from our lungs to our individual cells. So a good way to do that would be to have it bind to something that can then transport the oxygen to where we want it, but then it needs to be released. If it doesn't get released, that's a pretty useless way to transport oxygen. Okay, so when we're looking at our individual functions for our proteins, we want to evaluate kind of that dynamic situation. We want to be able to get our oxygen off in this case. So we use the protein because the heme works too well. So the protein is actually there to prevent oxygen from binding perfectly, to kind of disrupt that situation so that we can get the oxygen both on and off. Okay, so we need that off dynamic as well. The heme is often referred to as a prosthetic, just in case that word pops up. I think I'll end up making up stuff in a little bit. After that, we move up to our quaternary structure. So we briefly kind of end with looking at myoglobin. I will mention that our myoglobin is really not a very good transporter. Its primary function is actually storage. Stupid pen keeps rotating. So it's really a storage molecule. So if it stores oxygen, where would we want to find it in our body? We want to find it in the muscle tissue, okay? Because what happens in our muscle tissue if for some reason we don't get enough oxygen transported to it via our other proteins? Okay. We could ultimately die. You guys go through the nice not dying steps first. You go through, the anaerobic process. You can build up lactic acid and other things. Okay, so if our bloodstream isn't working appropriately or our lungs aren't working appropriately, but we still need to get that motor function, that's when myoglobin can come into effect and it can release that oxygen. So at very low oxygen pressures, it releases the oxygen and it can kind of give us a little delay factor to hopefully get some more oxygen from somewhere else, right, before we initiate into those other processes. When we move up to quaternary structure, what we're now looking at is proteins on top of proteins. So we're taking multiple different proteins in their folded state and we now combine them together into a larger, potentially more functional protein or potentially just functional in general. So for instance, our ATP synthase doesn't really function at all until all of the proteins come in. So we need all of those individual pieces. Okay? You can look at it as like the parts of a car. An engine's great, but an engine's not going to make you go anywhere if all you have is the engine. Okay, we need all those other pieces put together on top of it. Okay, so that's our quaternary structure. The scale up when we're looking at oxygen or oxygen interactions in our body would be hemoglobin. So hemoglobin is that structure shown on the left. And you'll notice it's kind of similar to myoglobin, and that we do have that heme located in the middle of each of those subunits. But what we're now looking at in the case of the hemoglobin as opposed to myoglobin is I need 4 protein pieces, okay, to come into it. We can add extra fancy words into this. It'll sometimes be referred to as tetrameric because tetra means 4. Okay. So all we're saying is that those things can now interact. One of the side effects of this interaction is that as it binds, potentially comes in, isn't going to change the interactions within each of those subunits. Right? Possibly yes, possibly no. So that's where we can get this new kind of side effect out of this. Okay? A peptide may affect another peptide. Right? That's referred to as an allosteric effect. Right? We'll talk about how we can see that mathematically in chapter 6. But what's happening is we can get those allosteric effects. Our structure can affect another structure. Hemoglobin does show this effect. Okay? And its primary characteristic is this thing known as positive cooperativity, meaning when one oxygen molecule binds, so if we take our hemoglobin with no oxygen, and an oxygen molecule binds, that causes a shift in this subunit. Okay? The shift in the subunit now changes how it interacts with the other subunits. Okay? Well, that change now changes what's around the heme in that other subunit, exposing it. Well, if it becomes more exposed, what can it do? It can grab another oxygen. That change further changes the structure, and it alters another subunit. Now that other subunit also changes its configuration and exposes its heme even more, which now means more oxygen can bind. So what we're saying is positive cooperativity. As we add an oxygen molecule, we actually facilitate the ability to bind more oxygen to that structure. Right? And it's that allosteric effect, the changing in the structure, that's now affecting the function, or the effectiveness of the function of each of those. In this case, that's your positive cooperativity for hemoglobin. Both of them are used to bind oxygen. So what we can do is take a look at some graphs. Is we've got two curves here. Okay? One's for our hemoglobin, one's for our myoglobin. Okay? If we take a look at those two curves, both are used to bind oxygen. What information can we interpret from these two curves? So we could look at saturation amounts. Okay, so the saturation on our y-axis is how much oxygen is actually bound into the protein. And you'll notice that the myoglobin starts to flatline about 100%. Why would it flatline?
Well, it can't be
It can't get any higher than 100%. So eventually it'll flatline. Your hemoglobin will eventually do that too, but we have to get to much higher pressures of oxygen to allow that to occur. So what we can do is pick some saturation levels, say our 50% saturation, and look at the pressure of oxygen necessary to get us to that 50%. Okay. And if we take a look at myoglobin, to get to 50% saturation, we're looking at very little pressure. Very little oxygen needs to be present, and myoglobin will hold that oxygen. Hemoglobin, on the other hand, we have to get to a much higher pressure. What might that tell us about the individual function of those? More efficient at what? Holding oxygen. So if we're going to look at our potential functions between these, we already talked about what myoglobin was. It holds oxygen very, very, very well. Okay? So if we think of what we need oxygen for, we need oxygen to get to our cells so that our cells live. So we get big because we have a bloodstream. Okay? If we're tiny unicellular organisms, or even slightly larger than unicellular, they don't have a bloodstream. Why do they not have a bloodstream? Every cell is exposed to oxygen. They don't need it. Right? We need it, so we have these two potential proteins that can interact with oxygen and potentially hold it. Right? And if we're looking at what happens is we inhale oxygen, makes it into our lungs, and then we need to get that oxygen to somewhere else, say our big toe. Okay? To get there, which of those two proteins is going to be the best option? Okay, why the hemoglobin? It will allow for transportation of the oxygen to get from one place to the other. The myoglobin is going to hold on to it and never let go of it. So if it was involved in, say, a transportation mechanism and actually moving through our bloodstream, all it's going to do is hold the oxygen. That's pretty useless. Okay? particularly if all it's going to do is hold it up at the heart, and eventually as it moves further away, we're going to start to, or not at the heart. What holds oxygen? Lungs, at our lungs.
Same vicinity.
Close enough, right? Okay. As it gets further away from the lung or our oxygen source, that's when our oxygen gets released. So our extremities end up with very little oxygen, but our core ends up with a lot. Well, it doesn't allow us to grow very big. Okay, so what we would need is something that can allow for a little bit more transport of that hemoglobin, a little bit less tightly bound, okay, or less tightly saturated. Okay? So what we're looking at as our primary function difference is that myoglobin allows for storage, and our hemoglobin allows for transport. And we can pick that information up off of the curve, just looking at how those two curves differ from each other. Right? Another thing that we might notice is that if you look at the hemoglobin, its curve doesn't move like the myoglobin. And I know this sounds a little bit weird, but we're going to attempt to show this. Hemoglobin could still potentially have the same shape curve as myoglobin, okay, just with a steep or a less steep binding curve. It could just go kind of flat and then level off. You'll notice it doesn't go flat. It doesn't have the same shape as our myoglobin. Our myoglobin is pretty much straight up and then starts to turn, okay, which is hyperbolic. Okay. Our hemoglobin does not have this hyperbolic situation. Okay. What this is referred to as our sigmoidal shape, because it looks kind of like an S. At least that's how I look at it. Okay, so we've got that kind of sigmoidal shape. Okay. And you'll notice that at relatively low oxygen pressures, we don't get any binding. But as we slowly increase the oxygen, we start to increase the binding. And not only do we just move linearly upwards, but it becomes much steeper curve. So at low oxygen pressures, there's virtually no binding. As we increase, you can't really see those lines. As we increase our oxygen pressure, our saturation, you'll notice that our binding curve becomes steeper and steeper. Okay? So, and that's the natural aspect of that curving shape. So if we kind of zoomed out, to have that curving, how can I draw this better? Maybe if we just go completely parabolic. At that bottom point, there's virtually no binding, so we can look at the tangent. As we start to increase, what happens to our binding? Okay, it increases until it levels off again. Okay. Well, what happened with our hemoglobin? What was one of its characteristics? When a single oxygen bound, what did it do? Change the structure to do what? To accept more oxygen and to facilitate oxygen binding. Right, so it's not just one oxygen binds and another oxygen binds and another oxygen binds, because that's just a straightforward linear curve. Okay. What's happening in this case is that when one oxygen binds, it changes the structure to facilitate more oxygen binding, to speed the process up. So what happens? We're going to see this kind of sigmoidal shape on it. That's a characteristic of the positive cooperativity. Okay? It's now facilitating more oxygen binding. Kind of a neat little process that we can pick up by looking at the curve and noticing the shape of the curve. Okay? The other thing we'll find is that our hemoglobin binds oxygen in a heavily dependent way on a lot of other properties. H+, CO2, BPG, Cl-. All of these things change how hemoglobin can bind oxygen. Why might we want that feature to show up? Kind of. What was the role of our hemoglobin? Transport. So in one part of the body, we want it to absorb oxygen like crazy. In the other part of the body, we want it to release oxygen like crazy. Okay. If we look at just its binding affinity for oxygen, we can see some of that dependence, so that when we move to different areas where we have less oxygen present, we can dump some of that oxygen off into our muscles. Okay? But we want something that's more, so that when our hemoglobin gets from the lungs, where it's fully saturated with oxygen, to now someplace that has no oxygen, we want to make sure that all of that oxygen is dumped. We don't want to leave any of it behind, because if we leave it behind, or if we leave it on the hemoglobin, then it didn't transport it. It's just carrying it around. So what things could we do to help dump it faster? We can start changing the structure. If we can change the structure of our hemoglobin, that can potentially push the oxygen off faster. Well, what things could control our structure? And in an easily reversible fashion. Easiest chemistry. You did it in Gen Chem, you did it in OChem. We've done it in Biochem. We got a whole chapter on it in Biochem.
It's oxidation.
Oxidation is stupidly hard in my opinion, but if you find it easy, then good. That's awesome for you. It's even easier than that. And as a hint, there's something drawn up there already that alludes to it. The what? The pH, which is a representation of what chemistry? Acid-base chemistry. It's easily reversible. Oxidation and reduction actually requires a significant change in energy. While it is reversible through oxidation and reduction, that does require extra steps involved with it. Acid-base chemistry is relatively straightforward. Right? So what the body can do is use a pH gradient in either the muscles or in the lungs to further alter oxygen's binding. Okay? So if we change the pH level, that can then interact with our protein, change its structure so that it either spits off or it takes on oxygen. So we can see an H+ dependence via pH. What else might we see? CO2. Why would CO2 affect our binding? What's one of the byproducts of cellular respiration? CO2. Where are we going to have a large concentration of CO2 released? All in our muscles. Okay? So if we have a higher concentration, say, in our muscles than anywhere else, we won't have as high a concentration in our lungs because we've expelled it. Okay? We can also look at CO2 as a concentration gradient and see if the CO2 structure can affect that. Okay? The CO2 is a little bit indirect. Where's the CO2 also factoring into? What's the buffer system used in pH in our bloodstream? Bicarbonate, which is the relationship between H+ and CO2. So both of those have a high dependence on our binding affinities. BPG is a small molecule. Bisphenol glycerate. Did I do that right? I think that's right. Small molecule that we'll talk about in a little bit more depth probably on Thursday. That's going to affect, how our proteins can bind that oxygen. So depending on what we're trying to do, we're going to look at different situations. So we want all of this dependence because of the role of our protein. What is it trying to do? Hemoglobin is trying to transport oxygen from one location to another. Myoglobin's role is simply to hold it. Okay? So that in times of dire need, it can then release the oxygen. We don't really want to have that as a store or as a transport mechanism. Okay? So our proteins have kind of self-evolved to give us better transportation for oxygen because we can now exist longer. If we had myoglobin as our sole form of transportation, okay, we're probably going to go through a lot of sprint systems, okay, because we have to get to very low concentrations of oxygen before it actually releases that oxygen. So we'll have a lot of anaerobic activity, and we can probably only do short sprints before we're now winded and say, okay, well now we've got to sit and relax. Now my oxygen level's back up. Now I can keep going. Okay? Hemoglobin, we've got this nice fun system where we have all of those extra factors that relate into it. That allows for the oxygen coming on and off. One of the big reactions that we could look at is this one at the bottom. Extremely helpful for some of the questions in the homework. So what are we looking at here? We're looking at hemoglobin bound with oxygen. When we add H+ and CO2, what happens? That arrow usually tells you what happens when you mix those three things.
reversing?
It releases oxygen, and we have our H plus and our CO2 now bound to our hemoglobin. Okay? So both of those things can affect the structure. So what happens if I increase the concentration of CO2? Drive to the right. So if we look at our muscle tissues trying to function and they release CO2, what happens? Hemoglobin floats through the system or floats by and goes, oh, CO2. What happens? Spits off the oxygen, so the oxygen can now be used in our aerobic respiration processes. What happens if we increase the pH? What was that, Jay? If we increase the pH, this one's obnoxious. Increase the pH does what?
Releases the hydrogen.
pH 7 is what? Neutral. If I increase the pH, what am I doing to the H plus concentration?
Decreasing it.
Decreasing it. If this decreases,
It's going down.
it shifts to the left. So you got to watch out. pH is an inverse relationship to H plus concentration. It's a very easy place to get tripped up. Okay? So you increase the pH, this concentration goes down, shifts your equilibrium to the left. Okay? Where might we find an increased pH? Why in the muscles?
Because it's trying to get rid of carbon dioxide.
So if we increase the pH, this concentration falls. Which way does the equilibrium go?
To the right.
To this side.
Yeah.
Which means, what is hemoglobin doing?
It's holding.
It's holding oxygen. Where do we want that to happen? In the lungs, not in our muscles.
I thought that one of the first lectures, didn't you say that the lungs had to be acidic?
I don't, that's going to be a long time ago.
I read that. The lungs had to be acidic.
What we're looking at is strictly based off of this equation.
Okay.
Off this equation for sure. What we're looking at is dropping that pH, or sorry, dropping the H plus concentration is for sure going to result in oxygen binding. Right? If oxygen is bound, that's got to be happening in the lungs, not in the muscles. Yeah, it's possible I switched it earlier. Without having an equation, it makes it more challenging. Okay? So what we're looking at is that relationship. To identify this, a lot of the questions in Sapling are going to come back to looking at this equation and changing each of those variables. What happens when you change the variables? That's going to change those relationships and cause different things to shift. Make sure that you're watching out for pH questions, because pH gives you an inverse relationship. Okay. Another situation, I know you may not have noticed that the slide just changed, but it did. If we take a look at the graph on the upper left, you'll notice that we've got a new graph here. We've got fetal hemoglobin versus maternal hemoglobin. And you'll notice that we have a slightly different curve for this. Fetal hemoglobin actually has a higher, a lower pressure to achieve 50% saturation. Why might that be the case?
You need more oxygen.
Fast twitch versus slow twitch muscle.
I wouldn't even get into that either. I think it's easier than that. What does the fetus breathe?
Fluid.
Fluid. It doesn't. So what does the mother need to do? Breathe both for herself and for the baby. The baby's going to have a lot more things going on and need more oxygen to deal with all that cell division and propagation, all that fun stuff. If it matched exactly, okay, you're getting equal concentration of oxygen to both the mother and the baby, but the baby arguably needs more to deal with all that cell division, okay, and all the growth. Okay. If it was lower than the mother, then what happens?
It wouldn't get as much.
The baby gets even less oxygen than the mother. That's even more problematic. So what ends up happening for your fetal hemoglobin it has a much higher binding affinity for oxygen. Because what it's trying to do is act like a parasite, and it has to, I mean, it didn't have much of a choice, and pull oxygen away from the mother so that it can survive. It has no other way to survive. Okay? So we would expect the binding curve for fetal hemoglobin to be a steeper curve than for the mother, because it needs to get that oxygen away from the mother.
Doesn't baby not need as much storage? There's a constant supply of oxygen, so the myoglobin supply overall is lower.
I don't know anything about that, so that's quite likely because it doesn't need that storage mechanism if it's constantly being supplied. So that would make sense. But we're looking at hemoglobin, so I can't answer that one. So that is very possible. I don't know.
Not to go backwards, but
Yeah.
to answer her question, you did talk about that when you're doing blood buffering. So on the slide, pH in the lungs, pH decreases, which draws CO2, and then in the muscle, pH increases, which deters CO2. That's what you said.
I need to see the equation. We can talk about it after class.
Yeah, it's,
To look at,
in case
because I think I know what you're talking about, and I think I know where that figure is. I think I can find
Where
it.
did it come from, where I got that in this one?
Yeah, it's for sure looking at the buffer system, so we can pull up that equation and see what it looks like. It's going to be a slightly different system because when we're looking at hemoglobin, hemoglobin's going to act as almost a buffer as well because it is pulling H plus off. So you really have two buffer systems. You have the CO2 buffer system, which is just your blood buffering, and then you also have the hemoglobin buffering system. So we're going to have slightly different systems happening in each of those cases. It's how both of them interact.
I just, when I remember something like that, I just need
No,
to make
that's
it make
fine.
sense, so appreciate it.
That's why I would always refer to the equation as best as you could. I'm hoping that I can find a better reference for hemoglobin and myoglobin. I don't think I'm going to put in more slides, but what I'll do is try and see if I can't find a good reference to read up on to help you get some more exposure to that, because I don't think the book is quite sufficient enough for what you will probably end up needing in future courses. Okay? I should have deleted this slide. This was actually based off an experiment. Okay. What could cause a protein to stop functioning properly? There can be all sorts of fun things that could cause this. Heat could cause it to stop functioning. Why would heat cause it to stop functioning? So this is where we got to be careful. It increases the energy, and it will break something. It very typically does not break bonds. It breaks my precious. It breaks the intermolecular forces. Okay, so we've got hydrogen bonding interactions, van der Waals forces. We've got all of these different interactions that are holding our protein in its native form, its fully folded state. If we add heat, we can now break each of those interactions. What happens when we break those interactions? Well, it's no longer in its native state. It's typically referred to as its denatured state. And so we lose all of that secondary and tertiary structure. So heat is something that could affect this. What else could affect it? pH, because pH is going to change the charge on your amino acid, which is going to change the interactions. What else? Solvent.
Yeah.
Right? We can just change the solvent in general. We can add in other things that will help break those interactions even quicker or stronger. Add in metal chelators. So add in a metal or an enzyme, or not an enzyme, a molecule that binds metal. Well, if something comes in and rips the metal out of the protein, well, the protein now doesn't have a metal ion to fold around. Okay, so pretty much any of those tertiary structure things that we talked about, if you can do anything to disrupt those, you're now going to push into the denatured protein. Okay? That's your primary difference. Denatured versus native. I should stop saying primary. It's our protein difference between our native and our denatured state. Okay? Do not break bonds to denature a protein. If you break bonds, you've destroyed the protein. It's no longer denatured. It doesn't exist anymore. Okay? So be careful with that. Protein folding, just because it's a pretty picture. What amino acids would we expect to be in red? Okay, where are the red ones versus the green ones? I know, it's not super easy to tell.
So the hydrophobic.
The red ones are all tend to be found on the interior in this case, which means they should be hydrophobic. Okay. All the green ones are found more typically kind of on the outside. It's an interpretation of a picture, so don't worry. Okay. The green ones are found more on the outside. Those are going to be our hydrophilic ones, or polar ones. Okay. I'll see what I can find on hemoglobin and myoglobin and get that posted. And then, that's just an empty slide. Then we'll move into chapter 5 very quickly. That's the only slide on chapter 5. Then we'll do chapter 6.